Thank you. Thanks, Angela. And thanks, for coming. I'm glad we had a good turnout. This is awesome. So, so today I'm going to talk, the title that I gave was Asian carp, where are they going and how can we stop them? So I think what I want to do is just kind of cover some of the research we've been doing in the lab for, I guess it's probably been about 3 years now. I'm surprised at how long it's been going. And so it gives, I want to basically cover two projects. One is a little bit more advanced. One's a little bit more developed, and the other one's kind of coming along. And so, but the data are really, really cool, and it's really fresh. And so I really want to kind of get it out. Maybe. All right, hold on, bear with me here. Okay, we're good. Okay, so the main focus that we're going to talk about today is carp and Asian carp. But just to kind of set the stage for a lot of this, I really want to take a bit of a step back and talk a little bit about just aquatic invasive species in general, and recognizing the fact that a lot of the stuff that I'm going to talk about today kind of falls into sort of invasive species biology and kind of in some ways kind of transcends a lot of the stuff we're doing specifically with carp. Invasive species, as I'm sure most of you have talked about in different classes and things, are kind of spreading all over the place. Common problem, proliferating, and are no end of problems for different areas around the world. A lot of some of the classic examples, the top picture there being zebra mussels as they kind of attack a native mussel. But the reality is that a lot of the spread of invasive species that we're seeing, whether it be aquatic or terrestrial or anywhere, is fueled a lot by international trade, commerce, movement. I read a paper, or came across a paper a bunch of years ago looking at just the numbers of flights that are moving internationally between different continents. And it's kind of a staggering number, and just our ability to move around the globe, ship things, transport things around the world is growing like never before. And that's kind of the source of a lot of where we're going in terms of having problems with a lot of invasive species. The other thing to keep in mind as we talk about invasive species biology, and it kind of bears a lot of fruit in terms of what we're doing with Asian carp, is the fact that eradication after establishment is a lot more difficult than it is to prevent the introduction of invasive species into new areas. So once these kind of invaders show up, it's very, very challenging, if not impossible, to try and get rid of them. And so a much better strategy is to try and keep them from showing up in the first place and do what we can to prevent the spread and prevent their introduction. Part of the reason why we really are concerned about invasive species broadly defined, whether they be aquatic or terrestrial, is the fact that there's a huge economic cost that is associated with them, in addition to the costs associated with the economic impact, or sort of the ecological impacts and the ecosystem level impacts. It's a paper from a few years ago that kind of put some numbers onto some of these issues, looking at $1 to $5 billion annually spent on eradication and control efforts for introduced fishes. If any of you are familiar with the sea lamprey effort going on in the Great Lakes, millions and millions of dollars spent every year to try and reduce or minimize the impacts of sea lamprey on native fishes. And this paper pointed out there's about 88 species of introduced mollusks, resulting in $1.7 billion annually in control costs, a lot of that with, say, zebra mussels and water intake pipes and those kinds of things. So huge efforts, a lot of dollars, and once these things show up, it's almost impossible to get rid of them. The Great Lakes are, from an aquatic standpoint, particularly have a strong history, a long history of aquatic invasive species introductions. So this is a paper from a few years ago documenting the cumulative introduction of invaders in the Great Lakes going back to 1840s. And it's just sort of showing that over the years there's more and more things showing up. The total they got there is creeping up on 200 non-invasive, or sorry, invasive animals that have been showing up since about the 1940s. And when you kind of do the math on that, that's a new aquatic invader showing up in the Great Lakes or being introduced into the Great Lakes every 28 weeks for the past, 100 plus years. So there's a lot of things. The Great Lakes being a center for shipping, a center for activity, really large population density, a lot of things showing up there, and a lot of problems with invasive species in the Great Lakes in particular. Where I wanted to go today, what we're going to focus on is some of the work we'll be doing in the lab with Asian carp. And I'm sure most of you have seen the videos online of the carp and the jumping carp and those kinds of things. So we don't have to kind of go into some of those details. But the reality is they're here and they're established, but they're spreading, kind of, sort of. I mean, we'll go into this in a little bit more detail. But when you look at the timeline, these are silver carp, big head carp. There's 4 or 5 species of sort of Asian carp with common carp and black carp. And but we're going to focus on silvers and big heads, which are kind of the two new prominent species that we've been working on and kind of been grabbing the headlines recently. Asian carp, these silvers and big heads, around the 1970s, introduced into the U.S. for the use in aquaculture. So they're filter feeders, and so they're brought over to, they basically swim with their mouths open and eat anything that kind of, ends up in their mouth. And so they're brought in to keep, for aquaculture, to keep ponds clean. They do a very, very good job of that. In the early 80s, there was a flood in the Mississippi River, and the Asian carp escaped in the Mississippi River, and so in the 80s, they've escaped. In the mid-80s, they're found in the Mississippi River, and then they started spreading northward. But where this, I think, becomes really interesting is just sort of this spread pattern. And I guess the short version of this is it's not a stampede of Asian carp away from this epicenter down in Arkansas. There's sort of been this really almost peculiar kind of slow spread away from this epicenter. For example, 1991 and 1998, they're found in the Illinois River. So it took about 10 years to really move into the Illinois River. They kind of seem to be creeping up on going into the Great Lakes, but they're not exactly knocking on the, they're kind of knocking on the door, but not more in a literal sense than in a figurative sense. So their spread has been a little bit peculiar, and we'll get to that in a few more details. And the reason that their people are really concerned about this and some of the potential problems can happen, or I got them here, but again, it's kind of a laundry list of potential things that can happen. They spread rapidly, they're very abundant. Extremely fast growing. I mean, a one-year-old carp is disproportionately larger, I'd say, than a lot of the native fishes at 1 year old. Highly fecund, anywhere from 265 to 2 million eggs per fish in a spawning event, and they can have multiple spawning events in a year. And then again, we disrupting food chains, eating low on the food web. They eat sort of bacteria and phytoplankton and small plankton that a lot of native fishes don't actually eat. The concept of them jumping out of the water, which is, although kind of humorous, can be very dangerous, very negatively impact a lot of boaters or anglers or those kinds of things. And then they can get into the Great Lakes, impacts on the sport fisheries, impacts on the non-sport fisheries, and the ecosystem in general. So these are the kind of things that they could potentially, some of the negative impacts they could potentially have on the ecosystem. But as I've kind of said, the reasons for their spread and why they're spreading or why they're not spreading, it's kind of unknown right now. We really don't have a lot of information or a lot of certainty in explaining why they're moving or why they're not moving around. For example, assuming these animals, right now they're in the Mississippi River, they're not in the Great Lakes, and assuming they do get into the Great Lakes, there's a great controversy now, a great debate on where will they end up? Where, will they continue all the way over to New York? Will they stay in Lake Michigan? Will they not go in Lake Michigan? No one really knows where they're going to end up. We don't really know what impact they would be or what impact they could have on either the lake or on the tributaries. We have a limited ability to collect them and sample them. They don't really recruit well into sort of traditional fisheries gear, meaning nets or electrofishing and those kinds of things. And so we really do need some additional information on what's going on, some of their potential impact and where they could find themselves. So with that kind of as the background, that kind of hopefully sets the stage for where I'm going to go today with the presentation. And the overall goal of the presentation is, I basically have two goals. There are going to be two separate portions of the presentation. The first goal in the first part of the talk is to improve our understanding of factors driving the spread and movement of Asian carp. So again, there's some of the spread is a little bit uncertain at this point, and where they're moving and how they're moving, and we could hopefully stand to learn a little bit there. So to address that first goal, first of all, I'm going to quantify landscape level variation in stress and condition and try and use that as a predictor for animal movement. And then a second part of that is to relate sort of small-scale habitat features like chlorophyll, turbidity, flow, species diversity to the health and the condition of the fish. So those are sort of the two parts for goal number one. And then goal number two is talk about some of the research we've been doing looking at alternate barriers to stop movements, okay? And we're using carbon dioxide. I'm having some pretty good success with carbon dioxide, hoping to share some of that with you today. So it's basically figuring out why are they moving, where are they moving, and how can we stop them, kind of the way we're looking at this. The second part is something we've been working on for a few years now. So we got a bunch of data on that. The first part is a master's thesis of Stephanie List, who's in my lab. And Stephanie's the data, she's just kind of crunching these data. Some of these are brand new, ooey gooey fresh. And so and she hasn't quite got to the second part of that question now, so I'm just going to focus on the first part now. So I'll show you some data on that first part, and we'll have to stay tuned for that second part a little bit later on. So spread and distribution then. So we can talk about this and where Asian carp are spreading and where they're not spreading. And again, I can look at this from a historical context. Right now, the leading edge, sort of the northern edge of Asian carp within the Illinois River is 45 miles south of Chicago. And the interesting thing is they've stayed about 45 miles south of Chicago for the past 3, 4, 5 years. They haven't really advanced past that yet. And as I've said, they were escaped out of Arkansas in the 80s or whatever. They really haven't moved north to South Dakota, Minnesota. They're not really showing up there. The Missouri River, they haven't really moved into the Missouri, the Wabash and the Ohio. They haven't really moved into the Wabash. They like the Illinois River. They're really dense in the Illinois River, but populations are really low in the Wabash, the Ohio, these kind of areas. They really don't seem to be advancing too far north. There's no reproduction. The most northern reproduction that we've been able to document is 135 miles south of Lake Michigan. And so again, we've got moving into some areas, not moving into other areas, reproducing in some areas, not reproducing in other areas. And so what's going on, what's driving this movement or not, the non-movement is really not clear. And improving our understanding of why fish are moving around, why they're not moving around is really important for if they do ever get into the Great Lakes, where could they end up and where would they not end up and some of the impacts that they might have. So we're really lucky here in Illinois because we're close to what we did for Stephanie's thesis was looking at 4 different populations or 4 different rivers because they have 4 different personalities and 4 different characteristics in terms of not only the physical habitat but also the carp populations within those areas. So the 4 rivers that we're looking at for here and what we're talking about here are the Illinois River, the Mississippi River, the Ohio River, and the Wabash, and they're on the map here with different colors: the Ohio, the Wabash, the Mississippi, and the Illinois. And the cool thing is that each of these different rivers have very, very different characteristics, okay? So the Illinois River, we're kind of doing our sampling kind of around here. We're not up in the northern part of the river. We're kind of down here. Very heavily managed, numerous backwaters, relatively nutrient rich, okay? A lot of nutrients coming down from Chicago. Mississippi River, and again, we're sort of in the southern part of this chunk of the Mississippi down here, so we get the input from the Illinois and from the Missouri. Very, very nutrient rich area, very large river. We have medium to high population densities. The Illinois River are the super high population densities, right? All the videos and all the jumping carp and everything I've shown so far with like exploding carp all over the water, that's all coming out of the Illinois River with super high population densities. Mississippi River, not quite as high population densities. The Ohio River is a very different river, very deep reservoir-like with low population densities. And the Wabash is very, very low population densities. And these 2 rivers here represent kind of the new invasion, the new invading front. But again, it's been about 30 years and the animals really haven't moved into this area, so we don't really understand why they have or why they have not moved into some of these areas. So what we're going to do for this part of the talk and for Stephanie's thesis is try and quantify the variation in stress and nutrition and condition of Asian carp at sort of these broad temporal, spatial and temporal scales, and look at how these populations vary across these scales, or across these individual rivers. And ideally what we want to do is look at the stress and nutrition data from these different populations and hopefully make some inferences about what could be driving spread of Asian carp or why they are spreading or why they're not spreading. And related again to sort of environmental variables, temporal variables, and then try and make some predictions about spread and distribution. So to do this, Stephanie got to spend the summer driving around all over Illinois and all on these rivers, sampling the data we're going to show here for this part is just silver carp, okay, so the jumping carp. And all the data we're going to show were fish that were more or less fish that just basically jumped in the boat and volunteered to be sampled. And once they jumped into the boat, we did what we call the grab and stab, where Stephanie would grab them, poke a needle, grab non-lethal blood sample from them. We did this on 4 different rivers, okay, so we did the Illinois, the Mississippi, the Ohio, and the Wabash. And in each of those 4 rivers were sampled across 3 different time periods. So we had midsummer, late summer, and then early fall. And then each of the rivers, we sampled 4 to 5 different reaches, okay? So small little segments. We had a river that we divided into 4 or 5 segments that we then sampled at 3 different time periods. Okay, so we got some spatial or some hierarchical arrangement of the data. And with each reach, we did about 10 different fish. Okay, so we had about 500 plus fish that we had sampled over the course of the study again to get this level of spatial and temporal variation. got the blood, brought it back to the lab, and we ran a bunch of different indices of health and condition to quantify how the fish were doing in these different environments. Our nutrition indices, we had 4 different nutrition indices that we ran from the blood: cholesterol, plasma proteins, triglycerides, and alkaline phosphatase. And these are indices that would respond to feeding or nutrition of the animal and be indicative of an animal that's been in good health and good feeding or recent feeding events and those kinds of things. We also measured stress in these fish, okay? So we measured cortisol, which is a stress hormone that's increased in stressful times. So it could be from lack of feeding, it could be from poor habitat. These kinds of things could relate to high stress. And then glucose is another stress index that we measured from the fish. We had this mess of response variables, and so to analyze the response variables, we use principal components analysis. And so principal components analysis, the point is to basically kind of find similarities and patterns in the different kinds of data. And we got really lucky with how things kind of turned out, because what we have over there on the far left are the different parameters that we measured. So triglycerides, cortisol, cholesterol, glucose, protein, and alkaline phosphatase. And the way the principal components kind of shook out is we had 3 principal components. Principal component 1 explained 28% of the variation in the data, and that's loaded heavily for alkaline phosphatase and triglycerides with a little bit of protein that kind of creeped in there. And so that's an index that describes short-term feeding. So those response variables all kind of respond quickly after an animal's been feeding. So that grouped out nicely that way. The second principal component that we had loaded exclusively for cortisol and for protein. And that then responds as an index or a component that describes nutrition and health over longer time periods, almost like an energy store, okay, as opposed to principal component 1, which is that short-term index that changes after feeding. Principal component 2 here is sort of health and long-term feeding and long-term nutrition. And then the third principal component was cortisol and glucose, which explained 18% of the variation, and that was an index of stress. Okay, and so we were able to divide our response variables into short-term feeding, long-term sort of nutrition, and then stress, and the way that things kind of played out. Then what we did is we sort of built sort of this global sort of analysis of covariance that looked at all the different variables that we collected. So we're looking at how the different principal components are going to vary according to these models here. So the complex variable that we looked at that really sort of drilled down into the depths of the data, we're looking at this particular variable, which was how these different principal components are going to vary across reach, or within the reaches and then the time periods. And we're recognizing that this is all nested within different rivers. Okay, so we can look at the rivers separately, the reaches and time periods. Okay, so really kind of getting down into the nitty-gritty of the data. But in addition, we looked at time period by itself, reach nested within river, and then just the river by itself, and then we looked at size variable as well. So that's how we looked at the different facets of the data. As an example, the way this would kind of turn out, this is the analysis of covariance for principal component 1, which is the short-term feeding index, and we can see some of the significance here. But let's not get mired in the stats and the numbers. We can actually kind of look at some of the data. When we actually start to look at the data and look at how those principal components vary across the different model components that we ran, there's I think some really, really cool stories we can look at. First of all, what we saw is that there's very little influence of that reach signature on any of the data that we looked at. There's very little influence on the short-term feeding. These are the data for the short-term. This is the data for the short-term feeding. But there's very little influence of that reach component on short-term feeding, on nutrition, and there's no influence of reach on stress. Okay. We saw significant differences, but there's really no trend, there's really no pattern, there's really nothing that jumped out at us. So what we have here, this is the data for short-term feeding. So higher principal component scores mean an animal that's recently had a higher index of feeding recently, versus the lower principal components, which is an animal that probably hasn't been feeding recently. The different colors of the time period, so this is time period 1, which is midsummer, late summer, and then early fall. Got the fall colors. There's the Mississippi River going from different reaches, going from upstream to the downstream reach. Okay, so what we have here is the most downstream reach across our 3 different time periods. And we see that there's basically, well, there's a significant change, but there's really no strong signal of how this reach is impacting the principal component. Sometimes we have poor feeding, higher feeding, poor feeding later in the summer. Sometimes we've got the inverse, where we've got good feeding, no feeding, great feeding. And so again, when we look at the reach signature here was a little bit messy, a little bit cloudy. Really nothing kind of jumping at us to say that there's something strong going on at the level of the individual reach. What we did see, though, which was kind of interesting, was some really cool stuff looking at how variation in some of these parameters kind of shook out across the different time periods. So now what we have on this is the stress index. This is cortisol and glucose, and the long-term feeding. Midsummer, late summer, early fall in the same kind of response. So we have a higher principal component. In this particular case, you have better feeding, better energy stores versus worse energy stores. And what we see is that across time periods, early fall had the lowest value for stress as well as the highest value for nutrition. So we start to see some of these inverse relationships. So animals that are well fed are going to have lower stress versus when you're not feeding, probably have higher stress happening within the individual. So we do start to see a time signature happening. And we also see a really cool pattern across rivers, which kind of shakes out as well. So here we've got the Illinois, the Mississippi, the Ohio, and the Wabash stress, and then that short-term feeding index. And again, what you see is that a lot of individuals have lower feeding. The Mississippi River, for example, has the lowest short-term feeding index, whereas here we have the highest stress happening in the Ohio River across all the different time periods. So the Mississippi River have the lowest short-term feeding. Ohio and Wabash did not experience reductions in feeding indices. Keeping in mind, again, these are the feeding, sorry, these ones here. That's that invading front. There's very low population densities here, so these animals are just starting to colonize this particular environment. Doesn't seem to be hindered by any kind of a poor feeding index. And then the Ohio River fish have the highest level of stress, and we're starting to kind of drill down into these and look at these in a little bit more detail. So in summary, then we can look at what we're seeing with some of these data. First of all, there's very little discernible trend in the reach scale variation. I didn't show all those data because we didn't have time, and quite frankly, the data were pretty boring, but there's very little index of anything happening at a reach or at a really small scale that's having any impact on feeding or nutrition or health or anything like that. But we do see that stress and nutrition across these different populations vary seasonally and also at the river scale. So fish from the different rivers are very different fish. There's very different things happening within a particular given year. In fall, feeding scores tend to increase and stress scores tend to decrease. And so maybe here potentially looking at a time when animals could be moving, really fat, really happy. Animals are potentially looking to have energy to move to new environments. Mississippi River had the lowest nutritional scores, both for that short-term feeding and the long-term nutrition. So the animals in the Mississippi aren't particularly getting a lot of food. I mean, it seems like a really nutrition-rich environment, but those particular individuals don't have great feeding scores. And then moving into the Ohio and the Wabash River, it didn't seem to be impaired by a food shortage. It's not like the animals living there had really crummy nutrition scores or anything like that. So it's not like they're limited by habitat or limited by resources that would keep them from moving into those environments. So a little bit, still kind of a work in progress with this. I think the second part of Stephanie's thesis, we're going to look at a lot more finer scale data. We're going to start to look at some community level responses, some metrics like species diversity, species density, fish densities, and these kinds of things, and kind of take this to the next step. But right now we can look at how things are kind of changing across this broad scale, the broad habitat scales, and try and get an idea of what's driving the animals to go to certain areas or not go to certain areas. So now, having talked about factors that are driving, moving and why they're moving, some landscape level variation, which is sort of why are fish moving or why are they not moving. Now we can focus on stopping movement. Okay, so now we understand a little bit, trying to get some conclusions about why fish are moving around. Let's try and stop them, okay, and some of the work we've been doing recently with carbon dioxide. And I think for people to really have an understanding and appreciation of why we sort of need to be stopping these fish, I think it's important to have, first of all, a historical context that's kind of focused on Chicago. And even though most people have been to Chicago, and most people have been to downtown and seen the Chicago River, and we know about the Chicago River and reversing the flow of the Chicago River and all those kinds of things. I think the concept of why people are doing that is often lost. This top figure here shows the hundreds or thousands of years ago the drainage pattern for waterways in the Chicago area. So city of Chicago, it's a little bit hard to see, but it's kind of where this white box is here. But historically, what was happening was the Mississippi River and the Great Lakes basins were separate, and the line of divide followed this yellow dotted line almost going right through downtown Chicago, meaning the Des Plaines River that flowed south went into the Mississippi, whereas the Chicago River up on the north part of the Chicago River there and the little Calumet River went into Lake Michigan. There was two separate waterways. The Mississippi River and the Great Lakes basins were completely separate. About 100 years ago, people got the bright idea to dig about 78 miles of canals, reverse the flow of the Chicago River, and start to connect the two basins. Okay, so what we have happening here is the Des Plaines River still kind of coming down, but now we dug the Cal-Sag channel that connect the Little Calumet River into the sanitary ship canal. Des Plaines River is kind of coming down, and the Chicago River, again, we dug some the Chicago area waterway system and the Chicago sanitary ship canal, connected these two, put some pumps. There's the Wilmette pumping station, more pumping stations kind of down here, pumping water down and sending water down the canal down into the Des Plaines River, into the Illinois River, and down to the Mississippi. So we reversed the flow of Chicago. We took the Lake Michigan water, pumped it downstream, and sent it all down the Des Plaines River. But the point now is, relative to where we were 100 plus years ago, there now is an open connection from the Mississippi River into the Great Lakes. So there is a mechanism, an easy channel, more or less, by which Asian carp can swim from, technically from Arkansas or Illinois or wherever you are, right into the Great Lakes. Why did we do this? Well, there's basically two reasons why this was done. First of all was to aid in the removal of sewage. Back in the late 1800s, Chicago was using Lake Michigan both to deposit its sewage and also to claim its drinking water, which is never a really good strategy. It's uncertain as to whether or not people were actually getting sick or whether it was actually just a threat of people getting sick, but they realized that we needed to change something, and so they decided to send their sewage down to down the Mississippi River. And right now, I think that the importance of that Chicago sanitary ship canal in terms of sewage disposal, I think, is undervalued. And it's estimated that during the winter, when there's low flows and low runoff, about 70% of the water in that Chicago ship canal is comprised of treated sewage effluent. And I think that's lost on a lot of people, is the role and the importance of that canal in terms of getting rid of the sewage. The other thing that happens is that the Chicago River and that Chicago sanitary ship canal is the plumbing of the city, okay? And stormwater and rainwater effluent washes into this river and washes downstream. And so without that outlet for the city, with so much impervious surface and there's so much runoff, there needs to be an outlet for a lot of that stormwater to run. The city of Chicago is growing up too large around the existing infrastructure of the stormwater treatment, and the city just can't handle it. There's a lot of combined sewage overflows, there's a lot of pumping stations, and a lot of that stormwater just gets pumped right into the Chicago ship canal. So there's two major functions with water quality and stormwater removal and sewage removal of that Chicago ship canal, in addition to transport of materials, the barge industry, the shipping industry, let alone recreation and leisure and these kinds of things. So this open connection between the Mississippi River and the Great Lakes Basin is actually quite important and quite necessary. And I think, you know, that kind of plays into this story in terms of Asian carp potential to invade the Great Lakes and those kinds of things. The reality is a lot of people are upset about this. And I'm sure most people have heard about lawsuits. Places like Michigan or New York or Pennsylvania launching lawsuits against Illinois to close in the ship canal. I mean, in reality, you know, when you kind of take a step back, you can solve this problem about Asian carp getting into the Great Lakes with 2 dump trucks full of dirt. You just kind of dump it in there, close the canal, and it's done. We have no more chance of invasion. But like you know, when you kind of think about the role of that canal, stormwater, sewage water, these kinds of things, it's not that easy to get to. Physical separation is being considered, if you guys are interested. This is a report that just came out, I think, in January, called Restoring the Natural Divide, put out by the Great Lakes Commission, showing 3 separate scenarios for physical separation between the Mississippi and the Great Lakes. So they got 3 potential sites, three potential scenarios for putting dams in the Chicago River for damming up and having physical separation. The price tag for that is in the cost of billions, depending if you want to do A, B, or C, someplace between $4 and $9 billion, and it's going to take someplace between 10 to 30 years to institute, if we were to start right now. So no short-term solution there. For a short-term solution to stop movement between the 2 basins, there's a single electric barrier, the electric on the Chicago waterway, that's designed to stop fish from moving from one basin to the other, which is an okay solution. It works great for large-ish fish, okay? Meaning adult Asian carp, 8 to 10 inch Asian carp works probably very, very well. where it doesn't really it loses effectiveness when you get into smaller fish, fingerling, fry-sized fish. The effectiveness of electricity, just basically due to physics, the electricity just passes around the fish. It doesn't go through them, and it doesn't stun them. And so small fish don't recruit well into that kind of gear. No barrier is 100% effective. Something will go through it. Hurricane's going to come through, knock out power for a week, and then we're sort of vulnerable that way too. So the existing electric barrier is effective, but it'd be nice to have something that would give us a little bit more safety, I guess. So that's where the project that we started to take on has come in. And what our goal was to do is to develop a non-physical barrier, chemical barrier that we could implement in the city of Chicago or elsewhere to act as a deterrent to slow or stop the movement of Asian carp. Again, something that would ideally provide redundancy, because no non-physical barrier is 100% effective. And so we could add something that would supplement the electric barrier to provide additional safety, some redundancy. Ideally, something that's going to target smaller fish and make up for the deficiency in that electric barrier. We also want to get something that's economically, politically, and environmentally feasible, meaning something that the public can get behind, and something that we can implement as part of a control prevention strategy to prevent the likelihood of Asian carp getting into the Great Lakes. And so where the rest of the talk then, I want to talk about the work that we've done looking at carbon dioxide and using additions of carbon dioxide into the water to act as a non-physical barrier to deter movements of Asian carp. Carbon dioxide is nice for a lot of reasons. It's easy to handle, low tech. People understand what carbon dioxide is. It's something they can relate to. You'd add carbon dioxide to the water and it causes, first of all, causes hypercarbia, which is just increased CO2, and it reduces the pH a little bit, and I got some numbers on that too, but so there's a small reduction in pH. Fish have CO2 sensors in their gills, okay, so it's something that fish in theory could and should respond to. And so what we wanted to do is look at whether CO2 can act as a chemical barrier to deter movements of carp. And we just had this paper come out a couple of weeks ago, so if you guys are interested, you can look at the data in another forum there. So to test this, we did some lab studies. Okay, so we've only really worked on this in the lab. And the setup we did a behavioral component, and then we did a physiological component. And both components looked something like this, where we brought fish into the lab. We looked at silver carp and bighead carp, but then we also worked with largemouth bass and with bluegill. So anything we did with the carp, we also did with largemouth bass and bluegill, which are native species. We basically had a surrogate for native fishes of Illinois. And so anything we're going to dump into the water, put into the water to stop the carp, would be cool to figure out how it's going to impact the native fish as well. So we brought fish into the lab, acclimate them to the lab, and we used basically buckets, and in this case these are the small buckets because it would have been for the bass and the bluegill, but when we had big buckets when we're dealing with the bighead carp and stuff. We basically exposed the fish to 3 different concentrations of carbon dioxide. We had a control, so 0 carbon dioxide, 30 milligrams per liter, and 70 milligrams per liter. So the concentrations aren't important, it's basically low, medium, and high. So expose them to low, medium, or high CO2. And we see there's the associated pH change. So the pH started around pH 8, and then as you add CO2, it forms carbonic acid, and so the pH drops. And so we went from 7 to 6.5. The behavioral part of the study, we put them in the buckets there we took the lids off, observed them for 3 hours. We looked at what we call ventilation rate. So basically it's literally how fast are their opercula moving, which is just a really sort of coarse proxy for how happy the fish are. And we looked at what we called irregular behaviors. That I'll get to in a second. Another group of fish got the same treatment, and then after an hour we drew blood from them, and we were able to measure some stress indices and sublethal stress indices, again to measure how happy or unhappy they are. And again, silver carp, bighead carp, largemouth bass, and bluegill. So for data looks something like this. So what we have here are irregular behaviors during that 3-hour period. Okay, so we're looking at things like surface ventilations of the fish, twitching, they would kind of start to twitch eventually, gill flaring, intermittent breathing, these kinds of things are what I mean by irregular behaviors, okay? So the y-axis there is the proportion of fish showing those irregular behaviors. We've got the control fish, the low and the high, so the low, medium, and high carbon dioxide exposure with the concentrations. Largemouth bass are black, bluegill are the light blue, silver carp are the red, and then bighead carp are the dark blue. And basically what you see is that just having fish in buckets, every now and again they're not unhappy, but we have a significant increase of irregular behaviors once we add carbon dioxide. So when we look at this sort of index of irregular behaviors, surface breathing, animals are kind of giving us the indication that they're unhappy being exposed to carbon dioxide. Yeah, up to 70 milligrams per liter. Then we looked at ventilation rates. Okay, so literally is how fast are they breathing, right? How fast are their opercula moving? So the y-axis there is just ventilations per minute. And these are the data only for the silver carp. The control are the dark blue, the low carbon dioxide are the light blue, and then the high carbon dioxide are the red. And what you see is if you just follow the dark blue bars, which are the control, if you put silver carp in a bucket and you watch them for 3 hours, their ventilation rates don't change. You add 30 milligrams per liter carbon dioxide, watch them for 3 hours, their ventilation rates don't change. We move up to the 70 milligrams per liter carbon dioxide, and we have control, and then after half an hour the red bars disappear, and that's because carbon dioxide is a fish anesthetic. And so the fish go to sleep, whether they like it or not. Carbon dioxide is an anesthetic, and we do stuff with fish movements and telemetry, and we need to knock the fish out to do surgeries to implant them with tags, and we use carbon dioxide. So carbon dioxide is a very effective, very strong behavioral response where if you take them up to 70 milligrams per liter, after about a half an hour, they're all belly up in their buckets. They're fine, they're just sleeping, they're just knocked out. So again, a very strong indicator that CO2 has a negative impact on the animals and that they don't like it. Then we can look at, again, some of the blood responses, and this is glucose, which is, again, is a stress indicator. So when you have higher glucose, you're stressed or you have a stress response. It's part of your stress response. Significant treatment effect here, meaning that after we have the control fish, again, the largemouth bass are black, bluegill, silver carp are red, and the big head carp are blue. And as we expose fish, we had to go with the 30 milligrams per liter, because if we do for an hour, right, if we go 70 milligrams per liter, they all pass out. So we do 30 milligrams per liter exposure for an hour, we have a significant increase in the stress response. And again, we have a whole suite of stress variables, and I don't have time to go into all of them, but they all show about the same response, where regardless of the species, they're all showing an increase in the stress response. The other thing we looked at were some molecular responses or indices of stress. So this is, it's a gene called HIF1 alpha, which is an index of, typically it responds to low oxygen. It often just is upregulated in periods of low oxygen, but in this particular case what we're looking at here is using it as an index of stress. But we were able to look at specific tissues. So these are only, they're the bluegill and the silver carp, it's 1 hour exposure to 30 milligrams CO2. Gill is the black bar, blue is the our heart, and then red is blood. And what you see is that, HIF1 alpha is upregulated for the bluegills in the gills and the heart, we're upregulated again in silver carp in the blood. So we're able to look at 1 hour exposure, seeing massive changes in the genetic response of the animal at the molecular level to being exposed to this particular, to CO2. And again, this is another stress gene. Heat shock protein 70, which is a molecular chaperone, and it stops proteins from denaturing, right? The proteins will lose their 3D structure. And again, for silver carp, again, we see this massive upregulation of stress genes in their hearts. So exposure to CO2 at 30 milligrams per liter for an hour starts to cause proteins in the hearts to denature. So it's really, the animals just don't like it, right down to their molecular level. They're showing a really negative response to being forced to be confined in this environment. The coolest thing that I think we did was we used this thing called a shuttle box to quantify the behavioral response of the animal. And a shuttle box is, this is a sort of the aerial view of this is the tank. And this is showing this side being pink and this side being yellow. And what this is a behavioral choice arena, computer-controlled behavioral choice arena, where we are able to manipulate water quality parameters independently in each side of this box. And so what we did is we take this box, just for the choice arena, filled full of water, throw a fish in there and it moves on to one side, it kind of hangs out. Whatever side the fish is in, we start to slowly increase concentrations of carbon dioxide. After a while, the animal becomes unhappy, it doesn't really like it, so it leaves and swims over to the other side. Whatever side it was on, we strip all the CO2 out, start adding it to the new side where the animal has moved to. Eventually it gets unhappy and swims back to the other side. So what we're able to do is, there's a camera over top of it, much like this, and we add CO2 to the system. we're able to basically chase the animal back and forth with CO2, okay? And we get multiple measurements from multiple fish of multiple species. We can increase our confidence in our data here. We can basically write down the pH or the CO2 concentration when the animal leaves. We can show that they'll actively avoid CO2. The data look fabulous. Largemouth bass, bluegill, and silver carp, and there's the concentration of carbon dioxide, where in this particular, first thing we measured was what we called irritation behavior. And basically, after being exposed to the CO2, the animals start to surface ventilate, they start to search around, start to get kind of woozy and kind of wobbly, and that happens at around 100 milligrams per liter CO2. They don't like it. Eventually, go a little bit higher at about 110, they eventually all leave. And if they don't like it, they try and be tough. If they try and resist and not leave, they pass out. So we win. But basically the data are great, no difference across species, and around 70, 100, 110 milligrams per liter, everyone takes off. So we're able to actually show an active avoidance of carbon dioxide in a tank, in a lab. And this is all fine and good to do in a lab in a small, I mean the footprint of that shuttle box is about the size of that table. All fine and good to do in a lab environment, in a really controlled environment. What about taking this out into the field? How easy is it to manipulate large volumes of water with carbon dioxide? And so we went to the pond facility we have here in Champaign on South Lincoln, where we have a bunch of outdoor ponds. We just wanted to see how easy is it to tinker with water at a larger scale in a pond. This is a picture of the pond here. It's a 10th acre pond of 0.03 hectares. It's about 6 feet deep. 427 cubic meters of water, approximate volume. Took some cylinders of carbon dioxide, took some air stones, threw it in the water. These are sondes, so these are water quality monitoring, little water quality logging devices. They measure pH. Throw 3 of these things in, one at the surface, one at the 1 meter depth, one at 2 meter depth, throw it in the water, throw on the CO2 and want to see what would happen. And the response of that volume of water to a little bit of addition of CO2 is almost instant. It happens so fast. Just dropped down to where we wanted it to be. pH of about 6.5, which is about 70, 80 milligrams per liter. This is time and hours of how long we were monitoring. The pH went from 7.5 or whatever it is down to the target that we wanted, because we wanted it to get to 6.5. Our goal was 6.5. Got down there instantly. This is the sonde that we had at the bottom of the pond. Sonde in the middle, sonde at the top. You see the CO2 eventually off gases and it kind of leaves. But the point is it happened really, really quickly. It's very easy to handle in the field. It's very, very easy to do. So to kind of wrap that up, small short-term exposure of fish to carbon dioxide, about 30 milligrams per liter for an hour. We see the animals reduce their ventilation rates. They have an elevated stress response, and the stress genes kind of turn on. So we have a molecular response, right? Proteins in their hearts start to denature and that kind of stuff. If we increase that exposure to 70 milligrams per liter, they start to lose equilibrium. And if we get it up to about 100 milligrams per liter with that shuttle box, they actively avoid it. They swim away. They'll leave, okay? And the cool thing too is that carbon dioxide is easily deployed in the field. It's easy to achieve those target concentrations, and it stays stable over really long periods of time. So it has really high potential to work as a fish deterrent when we deploy it in the field in a larger setting. And so that's all fine and good, but now we got some really cool ideas where we're going to go moving forward. So first of all, the question, when I've talked to a bunch of people like this or about this in the past, and the question that comes up obviously are things like logistics, right? And the reality is I don't have a good answer for you at this point. And by logistics, I mean things like, how much is it going to cost? If you want to do this in Chicago, what's that going to cost? I'm like, well, I don't know. How stable is it over time? Chicago area waterway is a flowing water body as opposed to a static pond. How do you deploy this? How is it going to impact mussels or minnows or, you know, the loch, all these kinds of things. But the reality is that we're making good progress on a lot of these kinds of things in terms of deployment. Deploying carbon dioxide at the scale of a Chicago River, that's just engineering. And the reality is I've talked with a lot of engineers that have done, I said, I want to get water this volume in the loch, and they just laugh. And they're like, yeah, whatever, we can do that. We work in the Ganges River in India. We're treating the Ganges River in India. Like, you know, or we work at wastewater treatment facilities where we treat billions of gallons of water a day. So you want to just do like a little lock, there's like, yeah, it's no problem. So the logistics of delivering carbon dioxide into the water, I think is easy. We still need to do research on, you know, we're going to drop the pH and what's that going to do to release of toxins, or how the change in pH can impact metal concentrations and these kinds of things. So we got to do some research on that. The cool thing in it, where I think this has the most potential, is to be a barrier against larval fish, right? And so this is, we just did this, many of you at the back might not even be able to see this, but there's basically a bunch of little fish in here. This is just a drinking water cup that we put a piece of screen on it, and people in the front might be able to see there's a whole bunch of little fish in there. We're able to do carbon dioxide exposures to fish that are little larval Asian carp that are bigger than your eyelash, but they're not much bigger than your eyelash. Because this is the size class of fish, if you recall, that can get through, or probably could get through the electric barrier. So if there's a weakness to the electric barrier, it's going to be a fish this size, assuming they ever get up to that point of the river. And so this is Clark Dennis's master's thesis looking at stress response and response of CO2 to eggs. He's done eggs, he's done larvae, and we're going to Wisconsin to work with the Fed folks to do some fingerling fish to see how susceptible small Asian carp are to CO2, because we had good work with good responses with some of the bigger fish. I'm going to field test this in a pond. Okay, so again, everything we've done so far has just been in the lab. The number of different things we can do. I was talking with basically a group of folks that builds pond aerators, and I said, hey, can you guys build pond aerators? I said, yeah. I said, hey, can you guys deliver CO2? And they're like, I don't know. We'll get back to you. And then they went away and they were like, yeah, we can deliver CO2. So they had this cool system rigged up to like have pH sensors in a pond, and we should be able to deploy CO2 in a pond, couple it with fish telemetry and monitor fish movements. And this is what they're doing up in Wisconsin right now with a bunch of different technology. So we'll end up going up to Wisconsin to work in ponds. And then we're probably going to actually field test this in a lock. We want to do this spring too, and take one of the locks on the Chicago waterway, Lockport Lock or something, and try and achieve target concentrations in a lock. And part of the motivation for doing this is that, lawsuits, people in Chicago screaming, the shipping industry, and they want, we're looking, a lot of folks are looking for sort of action, some sort of plan, some sort of like progress and things moving forward. And so we're going to try and do some stuff and try and get some results. So to wrap up, the overarching theme here that we're trying to address as we move forward with a lot of this research is that preventing spread of an invasive species is a better strategy than removal. Right? Once some unwanted guest arrives, it's very, very difficult to get them to leave. But if we can prevent them from moving, figure out why they're moving, why they're not moving, try and prevent them from moving someplace, we can have a greater probability of success. When we talk about some of the work that Stephanie's done, Asian carp show variability in stress and condition at those large spatial and temporal scales. The rivers are very different, the time periods are very different. The invading fronts don't seem to be negatively impacted. The Illinois River with the high densities of fish, there's really little indication of the animals being any kind of density dependence here. We do see an improved condition in the fall of the animals, but there's no strong patterns at the reaches within the individual rivers, so there's some river level variation going on. And also the carbon dioxide barrier, about 70 milligrams per liter, has potential to slow the spread of Asian carp, potentially all fish, right? There's a lot of bad things in the Great Lakes, like round gobies that are in the Great Lakes that are not in the Mississippi River Basin, that people want to stop from moving down. So I think carbon dioxide has a really good probability of acting as a barrier here to stop either through avoidance or possibly act as a barrier to stop animals from moving. So that's about all I had. Thank you very much, and I'm happy to take questions.
Check to see if CO2 causes premature egg hatch?
That we haven't done. That's something that would be really, really cool.
Killifish, for instance, if you've got ones that are ready to hatch, they will hatch immediately on CO2.
Huh.
Throw them across them.
That's...
So theoretically, you might be able to get them to hatch too early for them to be developed enough to survive.
Oh, that's a cool idea. Huh. That would actually be really, really good. Oh, that's a good one. Thank you. Jared.
What was your starting water for the lab experiment?
For pH?
Yeah.
Around 8.
Okay, because a lot of times in the upper Illinois River, the pH drops below 7, like naturally.
Yeah.
So I wonder if that would be inflated when you add CO2.
Yeah, so that's something we got to start looking at, is different water chemistry parameters, and how the good thing is we have a target CO2 concentration that we need to achieve, and I think it's just going to be a matter of adding more or less CO2 to reach that target concentration depending on the pH. But yeah, I mean we are looking at that, and we got plans to go around and basically have a water collecting party around the state.
Then you had like 100 milligrams per liter as the...
Where they seem to avoid, yeah.
What was the pH change from 100 milligrams per liter?
That's going to be around 6.5, maybe even down to about 6, but I don't think it's going to get lower than 6, about 6.5.
Because then you have the whole
Mm-hmm.
to worry about too.
Mm-hmm. Mm-hmm.
The river's so polluted.
Mm-hmm. Exactly.
Really concerned about that.
Yep. Absolutely.
So in the first half, you talked about you took all those measures to get a sense of
Mhm.
why they were moving,
mhm.
and I think I missed that conclusion. What do you get from that data about why they moved into some rivers and not others?
Well, that's kind of where we are right now, and hoping to be able to test that with some more fine scale variables, because you can almost argue that both ways. Like if they move, they could move because they're nutritionally rich, or they could move because they're nutritionally poor. Or they could move because there's lots of competition around, or because they're not. But I mean, the first step where we were was just, is there enough variability for us to be able to tease that apart?
And those reaches, because you are really focused on why there's no pattern and trying to make sense of whatever pattern may be there. Were those randomly sampled, those reaches? Like, did you pick points on the river that you...
A lot of it was based on the existing long-term data sets where they've been monitoring fish populations there for 10 to 15 years.
Okay.
And so we just went out with those crews, and so it's been a historical. We have good historical data too.
So does the historical data show there's differences in reaches?
Or
like, natural fish populations?
That I don't know, and I mean we won't. This is the first time that they've done it with the blood metrics, but in terms of the historical, I know that the Asian carp are becoming more abundant. So I don't really know though if the native populations are really showing a massive decline in abundance, but I know that the relative proportion of Asian carp to everything else is going up a lot. So.
Kind of a related question. You mentioned in the first half that the ones that you bled were ones that jumped out of the water, jumped in the boat or whatever. Is that a representative sample of the carp that are in that body of water, or are only the healthy ones jumping out of the water?
That's a really good question, but unfortunately that's one of the only ways you can get the fish, right? Because they get scared by motors. The motors scare them, and so they kind of take off. So unless we're going to start collecting by sale, is like the only other way we can try and get them. But I mean, yeah, I mean you're totally right. So.
Any good reason why they jump out of the water?
Don't really know. I mean, I think the best guess is it's a predator avoidance thing. If you look at them, a lot of times they have like sort of upturned mouths. They like to feed near the surface, and their eyes are kind of pointed down. So anything bad comes from below. And so if you're at the surface and something bad's coming from below, you just jump to get away from whatever's coming at you. So
what is the chance, even if we get an effective barrier, that some guy in a pickup truck and a trash
Absolutely.
can doesn't, undo it all?
Yep, absolutely. I mean, you're totally right. Either accidental release or intentional release.
I mean, that's always been a fisheries problem, right? People manipulating little lakes for their own,
Absolutely. Or even someone who's just disgruntled with the volume of money that's being spent and just like, we're done. I'm done. Here's dump, and it's over with. So
I'm kind of surprised it hasn't happened already. Or maybe it has and they just haven't taken it.
yeah, exactly. So, yeah.
Or your idea about limiting spread and the unwanted guest metaphor there. If the unwanted guest is in the house next door, limiting spread sounds like a good idea, but if he's in your house, then removal sounds like a better idea. Is there any strategies that are possible on that front? For example, some species have started to recognize zebra mussels or to adapt so that zebra mussels are a prey item.
Yeah.
Any sign that that's happening or could happen?
Not that I know, but I know that there is an extensive harvest effort that's been going on through DNR, and I can't remember, I think it was over 1 million pounds of Asian carp that have been removed, trying to turn it into, I know they're sending to a processing plant in Tennessee, so that might be going to like cat food or fertilizer or something like that, and I know there's meat being shipped to Asia. So that's happening, but whether or not it's happening naturally, I don't really know. And I think part of the problem is just so darn many fish that it's tough to make a dent in a lot of especially in the Illinois River.
We have one more question down here in front. There, yes.
So you had fish in pails when you're measuring, when you're putting CO2
Mhm.
in versus a control. The CO2 was for an hour. Did you measure the physiological response of the control ones after an hour in the pails?
Yep.
Before they were put in there?
Nope, they were simultaneously. Yep, and all the pails were aerated as well, so we can. There's some rumor kind of thing going around that CO2 will drive down the oxygen, and so the problem was not low oxygen. The problem was for sure CO2 exposure. Yep, absolutely.
All right, well, thanks for your questions. Let's give